Meiosis is split into two stages. You have Meiosis One and Meiosis Two. So the first part of Meiosis One is prophase. So what you can see here at the moment is that in the nucleus the chromosomes are uncondensed and they're also not paired up. So the first thing that happens is that the chromosomes condense and the centrioles move to the opposite poles. So here we have the centrioles at the opposite poles, and what you can see is that the chromosomes have condensed and they are now paired up, so they are in their homologous pairs, which we also call a bivalent. Crossing over can take place at this stage. The other thing that then happens is the nucleolus and the nuclear envelope disappear. And then we move into metaphase. Now at the end of prophase and the beginning of metaphase is when we see the formation of the spindle. So the spindle forms between the centrioles, so from pole to pole, and the homologous pairs, the bivalents, attach themselves onto the spindles by their centromeres. And you can see that they're in their pairs and they're in the middle of the spindle, so they're at what we call the equator. Once they've lined up, then the spindle, which is made up of microtubules, get pulled. So those microtubules pull on the chromosome and they pull them towards the poles. So one chromosome from each homologous pair gets pulled to the opposite poles. And you can see that they get pulled by the centromere, so the centromere is moving first. So this is anaphase, we've now finished anaphase. Anaphase is when they're pulled apart, and so now we have telophase. So the chromosomes are at the poles, the spindle disintegrates, the nucleolus, nuclear envelope reform, and then the cytoplasm divides and we get our two daughter cells, which are haploid. Now we move into meiosis 2. So the chromosomes recondense, the centrioles move again to opposite poles. They've also had to replicate because we originally just had 2 pairs of centrioles and we've now got 4. So the centrioles have replicated and moved to opposite poles. Yet again the nucleolus and nuclear envelope disappear. And then moving into metaphase we get the formation of the spindle. Now this time we don't have homologous pairs anymore in the cells because they were split apart during meiosis 1. So now each individual chromosome lines up on the equator of the spindle by the centromere. And now when we move into anaphase, the chromatids get pulled apart. So the centromere divides and one chromatid from each chromosome gets pulled to the opposite poles. Once they reach the poles, we then move into telophase. Again, the spindle disintegrates, nucleolus, nuclear envelope reform, and the cytoplasm divides during cytokinesis, and we end up with our 4 haploid daughter cells.